Hello, everyone. Thank you so much for inviting me to present our work in this conference. Today, I'm going to talk about advanced biofabrication of functional biomaterials for tissue engineering, regenerative medicine, and organ-on-a-chip applications. My name is Houman Savoji. I'm an assistant professor at Institute of Biomedical Engineering at University of Montreal. I'm also a scientist at Saint-Justin Research Center and transmitted chair in 3D bioprinting and regenerative medicine. So let's kick off this presentation with this story of Luke. At the age of 10, Luke was suffering from a neurodegenerative disease that led to different surgeries and malfunctioning bladders, and also made his kidney failure. So he couldn't enjoy the life with his peers, his friends, and he has to stop going to school because of this neurodegenerative disease. Thanks to tissue engineering, he got a new bladder made from his own cells that give his life back. And this is now Luke at the age of 27, that he's now wrestling coach at his school. He got back to school, he went to university, and now he's enjoying his life with his peers and friends. And he's healthy and living with new bladder that was made through his own cell by tissue engineering technologies. So tissue failure is a tremendous clinical burden worldwide. Approximately 5 million surgeries in 1 year are done in only United States to replace damaged tissue and organs. And this is the breakdown here. As you can see, 3 million for orthopedic and reconstructive surgeries, 1 million surgery for cardiovascular diseases, 300K for internal organs, 200K for neuronal diseases. And as you can see in this graph, the demand for tissue, demand for replacement of the tissues are increasing, and the donor supply is really decreasing or plateau. So there is basically a big gap between the big gap, or there is a big waiting list for people who are waiting to get the transplantation or tissue from a donor. So tissue transplant supply is insufficient, and the gold standard is using the synthetic implant that usually they fail from wear, fatigue, or biocompatibility. So we can really appreciate that there is an unmet need for tissue engineered graft and organs. Tissue engineering in general is replacement of the tissues by living damaged tissues or damaged organ by living tissues capable of growth and remodeling of the tissue. So in tissue engineering, basically we take the biopsy from the patients, and then we isolate the cells from this biopsy and the culture, and expand the cells inside the tissue culture plate, and then we proliferate these cells, and we combine these cells with the scaffold that we make from different biofabrication technology. And then we mature these cells using a mechanical, chemical, or electrical stimulation inside the bioreactor. And then after maturation, we implant these tissues to replace damaged tissue and organs in the body. There have been some success in the past for making different tissue, as you can see here, like trachea, ears, bladder, or different other tissues. So if we look at the last decade in tissue engineering, there have been different progress in different fields, in new materials, in novel chemistries, in biofabrication techniques like 3D bioprinting, or also like different, techniques to make this scaffold, like self-assembly technique or decellularization of the organs. And then in the cell side, we have different cell sources, invention of the human-induced pluripotent stem cells, genetic tools like CRISPR-Cas9. But there are still some challenges in tissue engineering that scientists are trying to address them. The first challenge, the first and most important challenge is vascularization. Vascularization is very important when you have a tissue that needs to be transplanted. This tissue should be stay alive pre-implantation and after implantation for host integration. So including or building a vascular network inside the tissue is an important task for scientists in this field. So first of all, we talked about macro tissues for implantation, but there is also another kind of application for tissue engineered basically grafts or even mini tissues or artificial tissues in drug discovery and screening application. So usually, if we look at the drug development pipeline, we start from a basic research. We have lots of candidates, drug candidates here, which takes 3 to 5 years to do the in vitro study, in vitro efficacy, in vivo efficacy, and then 5 to 7 years to go through clinical studies, phase 1, phase 2, and phase 3, and 1 to 2 years for approval of regulatory agencies like FDA, Health Canada, European Agency, and then one or two drug can get to the market and become available for the patients. And this is like between 10 to 15 years. And pharmaceutical companies contribute billions of dollars annually to bring 1 or 2 drugs to the market after 10 to 15 years of research and development and clinical studies. So, but most of these drugs fails due to the efficacy and safety issues, as you can see in this chart, and this caused the increased drug attrition rate. The most important, for example, that a drug failed after commercialization due to the efficacy and safety is the Vioxx that was designed for treating arthritis. And the side effect was cardiac toxicity. So this basically led to many deaths, and it was basically withdrawn from the market, and the company, the supplier and producer of this drug should pay lots of compensation to the family of the people who lost their life because of usage of this drug. So why this happened? Because usually during this in vitro efficacy assay and in vivo efficacy assays, we use Petri dishes, monolayer of the cells, 2D culture cells in a static condition, or we use animals, experimental animals, to prove the efficacy of the drug in vitro and in vivo. But none of these are really mimicking or representing what is happening in the human body, because our tissues in human body in human basically is are 3Ds, and basically there are basically differences between pathophysiology in animals and in human, and also genetic variation between animals and humans. So these models, experimental animals and 2D cell culture, are not really mimicking what we see in the human tissues. So then there is an alternative called organ-on-a-chip technology. Here in the middle that you can see, we can make this micro tissue or mini tissues using tissue engineering application, using patient-owned cells to make a reductionist model, which is inexpensive, with high controllability, and also high throughput, which is more also humanized using patient-owned cells or human-induced pluripotent stem cells to basically use this technology in drug development pipeline for drug discovery, screening, and disease modeling. So as you can see, this model is basically complement what we have in the conventional technology like experimental animals or in silico using computational modeling. So there have been individual organ-on-a-chip model that were presented in the literature individually, like lung on a chip, heart on a chip, kidney on a chip, gut on a chip, liver on a chip, and you name it, other tissues on a chip. And also this concept of body on a chip or human on a chip, that we can connect these individual tissues together to have interaction with each other and make a human on a chip model. That was basically proposed by the Wyss Institute in Harvard University. So in my lab, basically, we have different axes of the research, engineering new biomaterials, synthesizing and characterizing new biomaterials, and then making new tissue, new platforms for like organ-on-a-chip platform, and then for drug testing, for disease modeling, using different biofabrication technologies like 3D bioprinting, microfabrication, soft lithography, and make these micro tissues or mini tissues for disease modeling, drug screening application, and macro tissues for tissue and organ repair. So I wanna bring you a couple of examples of what we are doing in the lab. Basically, if we look at any tissues under microscope, you can see this fibrous structure. This is the muscle, so you can see the myofibers here. Basically, with 3D printing technology or 3D bioprinting technology, we can create these fibers. We can load these fibers with cells, and we can create different microfibers with multiple materials. We can weave them, we can braid them, we can stack them, we can make tissues thick, thin, and with different gradients. So this technology, biofabrication technology, including 3D bioprinting, enables us to basically make versatile artificial tissue using different cells and hydrogen. So there are different bioprinting technologies available in the market: extrusion-based bioprinting, inkjet bioprinting, and laser-based bioprinting. In the lab, we basically use extrusion-based bioprinting, sometimes inkjet bioprinting, and the focus basically is in cardiovascular diseases, which is a leading cause of death globally in the world. And the heart attack is basically a major cause of death in people. So the first project that I'm gonna describe for you here is making a small diameter vascular graft using electrospinning technology to answer or to address two clinical limitations. Basically, compliance mismatch means mechanical property of the commercial vascular graft are not matched with the mechanical property of the native tissue or arteries or coronary arteries, and then inadequate endothelialization means that the surface of these commercial vascular grafts are not suitable for cells to be adhered on these surfaces. So in this project, basically we look at the arteries. So the arteries or coronary arteries has three different artery has three different layers. The first layer is basically lumen or intima, the second layer media, and then outside is adventitia. And using electrospinning technology, which is coming from textile industry, basically we create these fibers and we collect these fibers on the mandrel. And then by playing with the parameters in electrospinning, which is voltage, the distance between the needle and the collector, the speed of the mandrel, we can have aligned or random fibers. Later, because these fibers are inert and they don't have any functional group for the cells to adhere to them, we make a coating called plasma polymerized coating to have these functional amine functional groups on the surface, and then we graft chondroitin sulfate, which is a sugar in the body, and bioactive, and then we culture these surfaces for the lumen side with UV to test and characterize the cell adhesion, proliferation, in a static condition and is under the flow or laminar shear flow, which is basically relevant for shear stress in the coronary arteries. So we were able to make these random or basically aligned fibers, and then we were able to have a monolayer of the UVEC on the surface of these electrospun scaffold. And as compared to conventional graft, woven graft, that you can see that it has a large pore size, so the cells go inside these pores. It is not favorable because in the lumen we have a monolayer of the endothelial cells that it's like a layer of the endothelial cells that are basically exposed to the blood here. So, but in the commercial graft, this is not possible because we have a large pore size and the cells go. So the live basically the adhesion and growth shows basically very successful results. So after the coating on our scaffold, adhesion and growth on both and aligned fibers increase over the time, over 21 days. And then the cells also are happy, they communicate with each other, and also they communicate with the scaffold. Cells on the aligned fibers, they have the random pattern on the random fibers, they have the random pattern, and on the aligned fibers, they have the aligned pattern. And also under the shear stress, 90% of the cells retain on the surface of the scaffold. It means that the cells are strongly attached to our bioactive coating under the 15 dyne per square centimeter, which is relevant shear stress in the arteries or coronary arteries. For the second layer, which is medial layer, we have basically, if we look at the native artery, we have these circumferentially aligned fibers that are included fibers plus smooth muscle cells. So we optimize our electrospinning protocol to have fibers with highly aligned fibers. Then we again functionalize the surface using plasma polymerization technique. We grafted chondroitin sulfate, and then this time to increase the bioactivity of the surface, we grafted also epidermal growth factors. And then we studied vascular smooth muscle cell adhesion growth, survival, and infiltration. So what we have seen is that the adhesion basically is increased over time, over 7 days, adhesion of the cells on the coated scaffold, and cells can penetrate inside the scaffold, because here by optimization of the parameter, we have a larger pore size that allows the cells to go inside and basically inside the pores and infiltrate inside the scaffold. The mechanical property of these fibers, we actually optimize it, and we, as you can see, are using plasma Asher or using etching technology can reach to the value of the femoral artery. So basically, to sum up, for more information, you can read these publications that are available basically on online. We address the compliance mismatch and inadequate endothelialization that are basically limitations in the clinic. And we made these multi-layer, two-layer, small diameter vascular graft, and we evaluated their performance in vitro. In the second project that I want to describe for you, we actually created a microfluidic printing technology with our colleague at the University of Victoria that enable us to have these fibers with different features like grooves on the fibers, inside the fibers, like hollow fibers, cross-section fibers, or fibers with multiple material, and we can braid them, we can weave them, and we can use this as cardiac patch, as steam patch. As you can see, the morphology, so we can have the different cross-section, like circular cross-section, or basically oval cross section, or cross-like cross section, and with the grooves that can be used as a clue for the cells, as a cue for the cells to attach and align through the grooves. So this microfluidic printing technology is a unique technique to make these microfibers in the lab. These are structures of the fibers. As you can see, the cardiac skin patch, the morphology and the porosity of the fibers, and also the woven structure that was shown here. And the cells also attached to these fibers, and they follow the grooves or micro grooves that we created on the fibers or inside the fibers. These are the cells, C2C12 cells, and these are the cardiac cells, basically cardiac fibroblasts and cardiomyocytes, that they follow basically the grooves compared to the fibers that they don't have a pattern or groove. So basically, we use these basically woven structures with these microfibers for cardiac patch or skin patch application. So in another project, so I created basically a microvasculature-on-a-chip using fresh printing technology, using a bioelastomer in the lab. Since we have a thick tissue in a 96 well plate, so we should have a microvasculature embedded inside or built in inside the tissue. So I created these micro, basically, vasculatures printed inside another hydrogel, and then install it inside these chambers, 96 well plate. And as you can see, this is the microvasculature structure, porosity in the wall, and also we endothelialize inside and inside this microvasculature. So we show the confluent layers of endothelial cell inside the microtubes, cross section, and then we see that thick tissue, cardiac tissue, containing hydrogel and cardiomyocyte. And as you can see, this tissue is beating in a long incubation time for 1 month. And can survive because of this microvasculature embedded inside. So, and also this platform can be used to connect different tissue. For example, cardiac tissue can be connected to liver tissue, and other things. So for more information, you can read this paper to get more information about this basically microvasculature on a chip. So in another project, we created artificial cardiac left ventricle using soft lithography technology. As you can see in the video, we created this scaffold inspired by the myofiber orientation from the heart that are changed from -60 to +60. So we created using soft lithography these basically a scaffold with micro channels, and then what we did is that we used this bioreactor that we created here to feed the cells on the top of this scaffold, and this is the cardiomyocyte cells, and then after that we wrapped this scaffold after maturation around this mandrel to make a mini heart in a conical structure that represents a mini heart. Here you can see the micro channel that we created, and you can see the cardiomyocytes that are basically are seated inside the microchannels, and they have the phenotype. You can see troponin C, alpha-actinin and DAPI that are expressed by cardiomyocyte. And then we have this micro mini heart that is able to pump a media. So here is basically our bioreactor system. This is our mini heart that basically after stimulation, you can see that the tissue respond to the stimulation. And for the volume change or pumping capability, you can see that it can basically pump 4%, 4%. The output, the cardiac output, the output that this mini heart has is around 3 to 4%. So then I switched the gear for basically sorting technology. Basically, when we are using stem cells, human-induced pluripotent stem cells, we use basically cardiomyocyte that are derived from the stem cells. Basically, we get the biopsy from the skin of the cells, then we program the cells with the iPSC cells, and then we get these iPS-derived cardiomyocyte. And when we see under the microscope, we see this beating sheet of cardiomyocyte. But usually, the population of these the cells are heterogeneous. We have endothelial cells, smooth muscle cells, and cardiac fibroblasts inside these basically cardiac tissue that is beating. So how we can sort or enrich the cardiac cells after differentiation? So usually we use FACS and MAX flow cytometry or magnetic cytometry to sort the cells, but we should have a specific marker. Usually there is no specific marker for cardiac cells or cardiomyocyte to be sorted by FACS. So one of the gold standard technique that widely used in the laboratory is pre-plating, because cardiomyocytes are non-adherent cells. So we pre-plate these cells. So cardiac fibroblasts, endothelial cells, or smooth muscle cells that are adherent, they adhere to the tissue, to the bottom of the basically flask. And the floating cells are cardiomyocyte, and we take them and we separate them. This technique is non-specific. Basically, it needs 2 rounds of sequential plating that takes time, and we reach to 80% purity of cardiomyocyte, and usually is non-consistent. So in a collaboration with University Technology Sydney, we use this spiral microfluidic platform for basically for label-free and high throughput cardiac cell sorting. using only centrifugal force and difference in size of the cells to separate non-cardiomyocyte cells from cardiomyocytes. So it has one entry to input to basically enter the cell population inside, the heterogeneous cell population inside, and two outlets to separate cardiomyocytes from non-cardiomyocyte cells. So we show that in the outlet, in one-time usage of this without any specific biomarker, only by using centrifugal or a spiral centrifugal force and a spiral basically force, we could get 90% in purity in cardiomyocyte population. And this video shows also the shows the cells that we basically plated cardiomyocyte, that they are viable after going through this platform in the outer and outlet of the platform. So this is also important. Another project in my lab is using 3D bioprinting technology for printing heart valves. So Arman Jafari, my PhD student in the lab, he created a new series of bioinks formulation using synthetic polymers or natural polymers, and then he printed or bioprinted this structure to make heart valves or tissue engineered heart valve for two specific applications. One, disease modeling application, and second of all, for transplantation. So basically, this is the design of the heart valve he used, and then this is the process of printing a heart valve in the lab without any support during the printing. And as you can see, he was able to create the heart valve with the leaflets. As you can see from top, you have distinctly three leaflets here. And as you can see in the micro CTs, he has all the feature of the heart valves, including sinus of valsalva. And the printing fidelity is excellent, and the biomaterial is biocompatible, cytocompatible, and we can see that we successfully created these heart to make sure that the material is biocompatible in vitro. We actually injected the material subcutaneously in mouse. After 3 months, there is no calcium deposition. This is very important for heart valve application because calcification is the major problem in the artificial or synthetic heart valves. And there is no evidence of basically chronic inflammation after 3 months. So this inflammation decreases after the 3 months, and we check different biomarkers, as you can see in this photo. This is unpublished data. Hopefully, when it's available, I can share the publication with you. In another project, another PhD candidate in my lab is aims to create 3D bioprinted heart on a chip for high throughput drug screening disease modeling application. Basically, he synthesized gelma and algma, and he mixed it with the electroconductive nanomaterials like reduced graphene oxide, and then he printed a ring-shaped structure using this material combined with the cardiac cells to create these biorings. So he characterized the hydrogel and confirmed the metacrillation of the alginate and gelatin. And then he measured mechanical properties of these material, different formulations, compared it with the native heart. So we are in the range of native. Young modulus is in the range of native tissue or tissue. He measured the compressive basically stress of the hydrogel, and basically after that he checked the printability window of the hydrogels, different formulations, rheological property assessment, and he found that the printing fidelity is great in the specific condition that he mentioned as shown here. So after that, he checked the viability of the cells during printing and after printing, after 5 days, with different concentration of the nanomaterials, reduced graphene oxide. The cells show higher viability. As you can see, these nanomaterials also are in the microstructure. Microstructure is really porous, and then the electrical activity also is increased by adding the reduced graphene oxide. He also assessed the swelling ratio of the material and degradation of the material. And then he aimed to print around, created this pillar structure, and then printed these biorings around these pillars to measure contraction force of the cardiac tissue. So this is ongoing work, and he's working to finalize this manuscript as soon as possible. In another project, Zainab Aji, my master student, created the thick vascularized cardiac tissue using GELMA structure. He actually printed this thick vascularized tissue, which is perfusable. As you can see, you can appreciate these channels inside the hydrogel, and she created basically this perfusable patch that can be populated with endothelial cells inside of these channels and outside with the cardiomyocyte that we are now doing the proof of concept with the human-induced pluripotent cardiomyocyte and human-induced pluripotent endothelial cells. So this shows that the patch is vascularized or perfusable, and then more investigation are being done now to use the induced pluripotent stem cell like endothelial cells or cardiomyocyte to show the functionality of this cardio patch in vitro, and then implantation in vivo in a rat, a model of myocardial infarction. So in another project related to the bone tissue engineering, basically we created these 3D printed PCL scaffold, combining these eggshell with the PCL pellet, and then we printed, and then we use plasma polymerization to activate the surface with the oxygen-rich coating. And then we showed that the cells really they can survive on this, and then they can breach between the fibers. We check the viability of the cells almost 90% after 20 after longer period of culture. There is we did antibacterial study because we use plasma polymerization and gentamicin grafting. So the antibacterial of gentamicin for bone tissue engineering application is 100% after 24 hours. And then we differentiate the cells, basically the mesenchymal stem cells. We differentiate it to the osteoblast. And here by PCR, we show that the biomarkers of that indicating the osteoblast differentiation are presented in these graphs. And alizarin red staining shows osteoblast basically generation from mesenchymal stem cells that is clear with these red cells. And these are morphology of the osteoblast on these printed structures. This work is already submitted, and once it's available, I can share the link with you. So in another project, collaboration with the University of Victoria, we created this biopen or handheld in situ 3D printer to print exactly on the damaged tissue like skin or heart using 4 cartridges. This is basically published in Biofabrication this year. So we can create different, fibers, multiple materials, different shapes, patch, or if you go a little bit creative, you can create with multiple material, different basically tissues, as you can see here. So we printed ex vivo on the pig skin. We also printed cardiomyocyte with these cells. So these fibers are functional. As you can see, these fibers are beating. And we can create multiple materials in one fibers. So the device is basically adaptable. It has a cooling and heating system, and also cross basically photo cross-linking feature on the tip of the basically pen. So this is an ongoing project. We are using this for different applications. We can create also these perfusable microtubes that can be embedded inside any tissue for making a vascularized thick tissue that can be implanted and integrated with the host tissue. So for conclusion, I can say that advances in biofabrication technologies enabled us to create complex biological structure with integrated vasculature at high spatial resolution. Although the clinical application of these engineered tissue at the moment are limited, but I can say that innovation in the stem cell technology, genetic editing technology like CRISPR-Cas9 technology, biomaterials engineering, immunomodulation, and 3D biofabrication can boost the clinical translation of engineered tissue. At the end, I would like to acknowledge the research funding that we received from University of Montreal, Saint-Justine Research Center, Montreal Transmedtech Institute, NSERC Natural Science and Engineering Research Council of Canada, TESEL Network, which is Quebec Cell Tissue Gene Therapy Network, and also FRQS, Fond Recherche Québec Santé, and also my students, my team, who basically are the main contributors to this work. Thank you so much for your attention, and I hope that you enjoyed this presentation. And if you have any question, you can always find my coordinates, my website, and email me if you have any question. Thank you so much again for inviting me, and it was a pleasure to present some of our work briefly in this Congress. Have a great time.